We were able to demonstrate that a decade ago with a microfabricated sample of which we had a scanning microscope image to begin with. We recorded the diffraction pattern, and to our delight, the reconstruction matched what we knew the right answer was. But typically in microscopy, you are not interested in imaging something of which you know what the answer is. And so we went on to try to look at some interesting biological specimens. And so we have specialized on trying to image yeast cells. This panel on the left-hand side is a logarithmic display of a diffraction pattern, which is, of course, full of speckles. And it illustrates some of the challenges that are involved in this technique. On the right-hand side, you can see the log scale of the counts of X-rays in the diffraction pattern as a function of the distance away from the center, which is the spatial frequency. And you notice that the dynamic range is 6 orders of magnitude, and that does not include that central black hole. This is not Andrea Ghez' black hole. This is a beam stop in order to protect our detector from the very intense radiation. And therefore, we have a lack of information at the crucial central point, which makes it more challenging to do the reconstruction. Nevertheless, we were able to do the reconstruction, and in fact, we had a bunch of diffraction patterns and a bunch of reconstructions near the, and can I go back? Yes, I can go back. Near the forward direction, but when I go back, there we go. So if I play this little movie, it shows you that all of these reconstructions are similar, and they give you a limited three-dimensional sense of what's going on. This year, we were able to image freeze-dried labeled yeast cells. The upper 3 panels show you 3 planes, the front, the back, and the center of the reconstruction. And on the lower panels, you either have the scanning micrograph or the scanning electron micrograph of the same cell. On the scanning electron micrograph, you can see gold labels on the surface. We can identify the same gold labels on the reconstruction of the diffraction micrograph, and from that we estimate that the resolution in this image is on the order of between 12 and 15 nanometers. Also, this last few months, we were able to get the first diffraction micrograph of a frozen hydrated yeast cell. The reason why we are interested in looking at these specimens in frozen hydrated state, because in the freeze-dried state, radiation damage impinges on our ability to get a faithful reconstruction, while in the frozen hydrated state, everything is frozen in place, so we can eventually get many images, so we can do a tomographic reconstruction at high resolution, but that will require a new beamline that we hope to build in the next few years. Now, ours is not the only way to do that, and Fourier transform holography is another excellent lensless imaging scheme, and there will be a talk on that subject also on Thursday at this conference. A very exciting development has been the development of tachography, which was proposed by John Rodenberg and then put into practice at the Swiss Light Source by Franz Pfeiffer and Pierre Thibault and others. And a member of that group, Andreas Menzel, is going to give actually 2 talks at this meeting, one tomorrow and one on Thursday. I think that this is really a very exciting development because it may make it easier to do the numerical reconstruction and does not require that the sample be isolated. And there are numerous other schemes, but I wanted to highlight the ones that are the subject of the meeting here. Another thing that has already been alluded to by Bob Bayer and several others is the advent of free electron lasers for X-rays. And the first one, the LINAC coherent light source, has already started operating and also has started doing experiments. And Jerry Hastings is going to give an invited talk about that, the first talk this afternoon. And there will be 4 other talks that I found in the program describing the various experiments that are either ongoing or planned using this machine. The Japanese free electron laser will come into operation next year, and the European free electron laser is planned for 2013. But at the same laboratory in Hamburg, there has been a vacuum ultraviolet soft X-ray free electron laser called FLASH that has been in operation now for several years. There are at least 2 invited talks on that subject at this conference. One of them this afternoon by Sorgenfrey et al., and the other one on Thursday by Anton Barty. And I will show some results from that. But before I do that, I want to sort of give you an oversimplified story about what's going on when you generate X-rays using electron bunches. Typically, an electron bunch has maybe on the order of 10 to the 9th electrons, and you put it through a magnet system which has one or many magnets. And if you just have one magnet like you have in an ordinary synchrotron, the X-ray output from a bending magnet will be proportional to the number of electrons in the bunch. If you make the electrons go through a bunch of magnets which bend it quite a bit, if you have n magnets, you can multiply this output by the number of electrons. And if you make the bends gently enough so that the photons and the electrons propagate together and interact, you can get an enhancement in the spectral peaks so that you go as the square of the number of magnet period. So that can give you a rather substantial factor. But the real increase comes when you go to a free electron laser, because there you micro-bunch the electrons so that the electrons are actually in a bunch whose length is comparable to the wavelengths of the X-rays that you emit. Just imagine you have 10 to the 9th electrons effectively within a few nanometers of each other. But under those circumstances, if you can do that trick, you get gained by something on the order of a factor of 10 to the 9th, and that's what free electron lasers do for you. So a free electron laser gives you fully coherent and very high peak power. And there has been a great deal of interest since the suggestions a decade ago that this could help structural biologists who are interested in solving the structure of protein molecules. If you can crystallize a protein molecule, well, that is a big business these days at all synchrotron light source to solve crystal structure of protein molecules. But unfortunately, a large class of protein molecules, especially membrane proteins, which are particularly important, refuse to crystallize. And so the idea was, can we get enough information using these very intense X-ray pulses on single molecules in order to get to the point where it is possible to reconstruct the molecular structure?